And now we're on to, excuse me, the subtopic of food ingredients produced by genetically engineered microorganisms. I repeat, food ingredients produced by genetically engineered microorganisms, and onto the subtopic of chymosin. A new food product is usually subject to a large battery of tests. And here, for example, the FDA approved the enzyme chymosin, an agent produced by recombinant DNA technology for use in cheese making without demanding a full range of tests. So one of the key components of RNA is a milk clotting proteolytic enzyme that hydrolyzes the casein protein of milk. This enzymatic cleavage creates curds, which in turn are processed into cheese. Traditionally, the milk clotting agent for cheese making is derived from the fourth stomach of carbs and consists of a mixture of food substances that collectively is called RNA. To ensure a reliable, convenient, and possibly cheaper industrial supply of chymosin, one of the chymosin genes was cloned and expressed, and the product was harvested from E. coli K12. And a petition requesting permission to use recombinant system for commercial production of cheese, when presented to the FDA, was taken with conditions that it was necessary to decide what criteria should be required for the approval process, because there's been a long history of using RNA containing chymosin in the cheese making industry. And in a sense, the petitioner had to show that the recombinant chymosin was identical to the chymosin of RNA. So to substantiate this restriction mapping, DNA hybridization and DNA sequencing were used to establish that the cloned and native DNA sequences of the chymosin gene were identical. Moreover, the recombinant chymosin had the same molecular weight as purified calf chymosin, and the biological activities of the forms of the enzyme were the same. It was essential to establish that the recombinant chymosin preparation was safe. The company showed that as part of its purification process, recombinant chymosin is extracted from inclusion bodies and that the final preparation is free of whole bacterial cells. After compiling all information, the FDA concluded that the recombinant chymosin could be licensed for commercial use. Currently, about 85% of all cheeses in the United States produce recombinant chymosin. Now, tryptophan. For the most part, agencies in various countries that are responsible for regulating food and foodstuffs derived from recombinant DNA technology have adopted a case-by-case approach. Each submission is considered separately, and depending on the judgment of the regulatory body, a series of tests is specified to ensure that the product is safe. Currently, the introduction of recombinant foodstuffs for human consumption is being handled with a degree of caution, especially since a false assumption that seems logical initially caused unexpected and perhaps tragic results. During 1890 in the United States, an unusually high number of cases of disease eosinophilia syndrome (EMS) were reported. This generally rare disease causes severe debilitating muscle pain and can be as fatal as a result of respiratory arrest. A consistent feature among the occurrence of EMS was that the patients had been consuming large doses of amino acid tryptophan as a food supplement. In each case, the source of the tryptophan was traced back to a single chemical company. The possible correlation between tryptophan and EMS was puzzling because there had been no history of significant negative effects when tryptophan extract from E. coli had previously been used as a dietary agent. Further investigation revealed that all of the suspected batches of the tainted tryptophan had been produced by a genetically engineered E. coli strain that had been designed to overproduce tryptophan. The company had assumed that the enhanced strain was identical to the previous one, therefore no additional product safety tests were thought to be necessary. At the same time, that was thought to be a minor step. What was thought to be a minor step in the purification process were changed, while the previous quality control measures that have been used to assay the purity of the final preparations were retained. Chemical analysis of the commercial preparations that had been produced by the genetically engineered strain revealed that they contain novel metabolic derivatives of tryptophan, including 1,1-ethyl ethylidene, bis-tryptophan, EBT. Initially, the presence of EBT was considered to be due to some metabolic quirk on the new strain. And while research focused on determining whether EBT caused EMS, other studies show that EBT was produced by wild-type strains as well. Toxicity studies established that EBT induced pathological changes in rats, similar to EMS, and surprising that even tryptophan to a lesser extent. And produced some EMS symptoms. Consequently, L-tryptophan, even without impurities, was banned for consumption in the United States. It's not clear what exactly caused EBT to appear in a product that had previously been safe, but most observers believe that the change in the purification process allowed EBT to contaminate tryptophan, presumably although the company was unaware of it, but the old method effectively removed it. And one of the lessons of this episode, even though genetic engineering may not have been the problem, is that biological equivalence between a strain and its genetically altered counterpart should not be assumed. This is as true for a strain produced by traditional methods as it is one that has been genetically engineered. Furthermore, manufacturers are now more aware that a minor technical change in the purification phase can alter the nature of a product. However, what they do with this knowledge may be problematic. Many companies do not want to run a complete battery of toxicity tests for a product that they believe has been thoroughly tested. However, despite economic costs, many manufacturers are opting for a better safe than sorry approach. Bovine somatotropin. The bovine somatotropin, also called BST, small bst, bovine somatotropin, or bovine growth hormone, controversy illustrates the constellation of issues that can arise from implementing a recombinant DNA product. In this case, Monsanto sought and won approval to market recombinant bovine somatotropin post-lac. Concerns about animal health and welfare, human food safety, and socioeconomic backers of small dairy farmers all came into play. Also, government agencies from different countries disagreed with some of the conclusions of FDA. Bovine somatotropin, also called BST, PSO, small bst, bovine somatotropin, or became bovine growth hormone. Controversy illustrates the constellation of issues that can arise from implementing recombinant DNA product. In this case, Monsanto sought and won approval to market recombinant bovine somatotropin post-lac. Concerns about animal health and welfare, human food safety, and socioeconomic impact on small dairy farmers all came into play. Also, governmental agencies from different countries disagreed with some of the conclusions of the FDA. In the 30s, it was shown that injection of bovine somatotropin into a dairy cow increased milk yield significantly, because natural bovine somatotropin is both difficult and costly to accumulate in large quantities. It has not been used routinely as an augmented agent by the dairy industry. However, with recombinant DNA technology, the gene for bovine somatotropin was cloned in E. coli and expressed. Recombinant bovine somatotropin harvested, purified, and tested under trial conditions. Milk production in dairy cows increased by 20-25% after injection of recombinant bovine somatotropin. The safety of natural bovine somatotropin milk has been studied exhaustively in treated cows. The levels of this bovine somatotropin milk are not higher than those in controlled cows. Moreover, bovine somatotropin is not active in humans at all toxicity rate trials has shown that there are no adverse effects on test organisms. The FDA, using all the research results that it could assemble, concluded that both the meat and milk of recombinant bovine somatotropin-treated cows are safe for human consumption. This conclusion was supported by the U.S. Office of Technology Assessment after its independent analysis of many of the bovine somatotropin studies on 5th of November 1993. Monsanto's application to use recombinant bovine somatotropin as a milk production answer was granted. An effective and powerful lobby group have been assembled to block governmental approval of recombinant bovine somatotropin. The principal reason for this opposition was based on the presumed economic consequences that recombinant bovine somatotropin would have on the dairy industry. Their fear was that many small dairy farmers would become unprofitable because fewer cows would be required to maintain current levels of milk production. In addition to the attrition of small dairy farmers, opponents of recombinant bovine somatotropin saw that the industry would become dominated by large corporate interests at the expense of independent producers. At the time, these economic arguments may have been justified, and certainly any group is entitled to protect itself against what perceives to be a threat to its livelihood. However, the main issue in the advertising campaign waged against recombinant bovine somatotropin was that genetically engineered hormones and hormone-laced milk would be harmful and cause cancer in humans. Using recombinant DNA technology as a bogeyman probably made this campaign emotionally effective. In addition to economic concerns, opponents of recombinant bovine somatotropin contended that its use would increase the incidence of bacterial infection of milk glands mastitis in dairy cattle. It was further argued that larger than normal amounts of antibiotics would have to be used to maintain the health of recombinant bovine somatotropin-treated animals that were resulting in increased levels of antibiotics in the cows' milk that might trigger allergic responses in some consumers. Moreover, increased antibiotic use could heighten the selection pressure for drug-resistant pathogens. The Veterinary Medicine Advisory Committee of the FDA and others have studied this issue and concluded that the frequencies of mastitis in treated and untreated cows are no different. On this basis, there's no reason to believe that the amounts of antibiotics in milk from recombinant bovine somatotropin-treated cows would be any greater than those from untreated cows. Moreover, by law, after any cow is treated with antibiotics, it is not milked for a specified period of time to enable the medication to be cleared from its system. The concern about this increased risk of cancer was based on the presumed increased concentration of insulin-like growth factor 1 in the milk of treated cows. The FDA maintains that the amount of insulin-like growth factor 1 in recombinant bovine somatotropin stimulated milk falls within the normal range observed for untreated milk. Moreover, any additional insulin-like growth factor 1 would add only a small amount in the existing pool of insulin-like growth factor 1 in human plasma. Taking into consideration these and other matters, the FDA reviewed the complete recombinant bovine somatotropin file in 1999 and found no reason to rescind its original decision. Notwithstanding the certainty of both the FDA and the Joint Food and Agriculture World Health Organization Expert Committee on Food Additives that recombinant bovine somatotropin poses no hazards for treated animals, the Canadian equivalent of the FDA Health Canada and European Union both refused to approve recombinant bovine somatotropin on the grounds that it is detrimental to treated animals In these instances, appointed committees conclude that the recombinant bovine somatotropin supplementation increases the risk of status, causes leg and foot disorders, decreases reproductive capabilities, and induces severe reactions at the site of injection. FDA analysis and more recent studies do not support these assertions. Clearly, the biological consequences of recombinant bovine somatotropin remain controversial. Currently, in the United States, about 15% of dairy producers use Posilac. There have been no reports since the onset of its commercial use that recombinant bovine somatotropin has had debilitating effects on treated cows. Studies also indicate that recombinant bovine somatotropin treatment has neither had an apparent impact on consumer prices, nor led to extensive consolidation of small dairy farms. Moreover, a new market niche has been created. In many food stores, the consumer is given a choice of milk and milk products from either recombinant bovine somatotropin treated or untreated cows. By 1996, many of the initial apprehensions about regulating foodstuffs that were developed with recombinant DNA technologies appear to have subsided in the United States as the FDA implemented traditional risk assessment procedures. The food industry preferred a minimal set of regulations to speed up the transition from developmental phase to the marketplace. In this context, governmental agencies as representatives of the public have a dual responsibility. They are entrusted with protecting the public health and ensuring that new developments are not needlessly curtailed. Presumably, any changes of regulations will not be made for sake of expediency at the expense of safety. Genetically modified crops. While there is still polarization on the issue of genetically modified crops, many national governments have approved the commercialization of transgenic plants. The United States cultivates more than 62 million hectares, which represents about 50% of the total global area planted with transgenic crops. In total, 25 countries now grow transgenic crops, with developing countries beginning to outpace industrial countries in their rate of increase. 4 crops, soybean, corn, cotton, and canola, represent about 99% of the genetically engineered crops, with squash, papaya, alfalfa, sugar beet, tomato, sweet pepper, petunia, and carnation making up the balance. Most have been engineered to increase productivity by incorporating genes that count for tolerance for herbicides and resistance to insect predation. Other traits that have been approved but are marketed to a lesser extent include resistant to viral infection, especially in papaya, and altered plant quality, extreme flower color in carnations. While recognizing the need to increase agricultural production, the overreaching purpose of regulating commercialization of transgenic crops, as for any crop, is to ensure that the crops are safe for humans and livestock to eat and safe to grow. Not only must the products be non-toxic for consumption, there must also be consideration for the potential impact on other organisms and environments outside the area of cultivation, because the plants are usually grown in an open field environment. For example, there is potential of plants carrying insecticidal toxins to adversely affect non-target insects or to crossbreed with wild plants, which could enhance their invasiveness. In many countries, regulations governing transgenic crops are still being developed and are evolving. In establishing regulations, the general consensus among national regulatory agencies has been to consider the characteristics of a transgenic plant rather than the process by which it is created. The prevalent view of transgenic plants have been that they are not different from traditional plant strains, cultivars that are derived from traditional breeding experiments. In the United States, 3 agencies are responsible for assessing and approving applications for for the development and release of genetically modified crops. The USDA is responsible for protecting the agriculture and the environment from pests. The FDA is responsible for the safety of human food and animal feed. And the EPA is responsible for regulating pesticides, including plants that are engineered to produce pesticides. The same testing and licensing procedures apply to all plants to carry genetic modifications, regardless of how these changes were introduced. Many other countries have taken a similar approach when adopting legislation and follow general guidelines provided by the Organization for Economic Cooperation and Development and the Cartagena Protocol on Biosafety, which was established to develop international standards for biosafety. The regulatory system in the European Union differs somewhat from that of the United States in placing greater emphasis on the process by which the crops have been developed. There is also a requirement for additional information that is used for labeling and to enable tracking of the origin of the plant. In general, each new transgenic plant is considered on a case-by-case basis. Even when a previously approved transgene is introduced into a different plant variety or in the same plant genotype, each transgenesis event is introduced in a new application because the site of insertion into the plant genome is random and therefore the potential for disruption of the function or regulation of an endogenous or introduced genes must be considered. Other traits that are assessed include the rate and method of reproduction, the potential for transfer of genetic material to other organisms, toxicity to other organisms, and sexual compatibility with wild relatives, for example, plants engineered with insecticidal proteins. Strategies to manage insect resistance must also be in place. To date, in the United States, 12,000 field trials of genetically modified plants have been authorized. This is the fourth edition, so that much of it could have advanced a lot by now. This is for basic information. Over 100 transgenic crops have been approved for commercialization. Some argue that the lengthy and costly pre- and post-commercialization regulatory process is prohibitive to the development of new transgenic crops with traits other than herbicide and pest resistance, which would be produced on a smaller scale. They also argue that the high cost of meeting regulatory requirements, estimated to be $20 million to $30 million per product, precludes development by anyone other than large multinational companies. Proponents maintain that after 20 years of scrutiny and incorporation of many of the safeguards, to ensure the transgenic crops are safe for consumption and release into the environment and regulatory process could be streamlined. Despite the successful history of transgenic crops, recent legal cases in the US may indicate that regulatory scrutiny would increase rather than be relaxed, as some producers had hoped. In 3 separate cases, it was ruled that field trials with genetically engineered turf grass in Oregon, corn and sugarcane in Hawaii, and commercial cultivation of genetically engineered alfalfa in California were approved by the USDA without adequate consideration of the environmental impact of the release. Somewhat more challenging is the regulation of transgenic plants that produce pharmaceutical proteins. Frequently, these agricultural crop plants such as corn, tobacco, potato, rice, and safflower, although so far they have been developed for production, contain facilities, for example, in cell cultures. Pharmaceutical plants grown in greenhouses, cell culture systems, and other contained facilities are regulated as drugs. The plants are genetically manipulated, often with several different genes, to maximize yields of proteins that are intended to be biologically active in humans or other animals. Thus, the plants potentially pose a greater risk to human health and the environment when growth in the field, and therefore are given special consideration by regulators. For example, There are concerns that material derived from plants could inadvertently affect the food chain, perhaps through seed dispersal. Prevention of such problems may require special confinement measures, such as dedicated farm machinery, containment netting to separate pharmaceutical crops from food crops, employing non-food crops such as tobacco to produce pharmaceutical proteins would circumvent some of these problems and also provide tobacco farmers who are facing some income losses due to a decline in tobacco consumption with an alternative market. Genetically engineered livestock. The regulation of genetically engineered livestock is not fundamentally different from that of genetically modified crops. Generally, the same agencies in the United States are responsible for regulatory oversight of both. As with cultivation of plants, the goal for the commercial production of animals, regardless of the method of production, is to ensure that the animal products are safe for use and that the impact of the animals on the environment is as low as possible. In addition, the well-being of the animals must be protected. For new, rapidly developing and expanding technologies, regulators are challenged with ensuring that these goals are met while recognizing the power of the technology to solve important problems in agriculture and medicine, and moreover, with acceptance of the products of these animals and other commercial viability, society must be confident that safety issues have been addressed. The regulatory issues for cloned livestock that have not undergone modification of their genomes are separated from those for transgenic animals. In the U.S. Center for Veterinary Medicine of the FDA considers that the cloning of livestock through somatic cell nuclear transfer is not different from other assisted reproductive technologies that have been practiced for many years and does not pose greater risks. Many cloned animals are unhealthy, most likely because the genome transferred from a different somatic cell fails to undergo epigenetic reprogramming. This normally returns the genomes of differentiated egg and sperm cells to a nascent state and an early embryo, which is required for normal development. However, sick animals are generally destroyed and their tissues are not consumed as food by humans or other animals. Healthy cloned animals are used primarily as breeding stock. The offspring which are consumed as food and are produced through sexual reproduction and therefore develop normally, that is, undergo proper epigenetic programming and are generally born healthy. The FDA takes the position that healthy animals produce safe foods. After assessment of meat and milk composition, it was found that meat and milk derived from cloned animals is non-derived and is also as safe to eat as that from animals produced using conventional agriculture practices. Additional regulations specific for cloned animals were determined to be unnecessary because any problems associated with the products from cloned animals can be identified through food inspections in current practice. All genetically modified animals must be approved before they are commercialized, and to date, no animals that enter the food chain have received regulatory approval. In the U.S., genetically engineered animals, whether containing heritable or non-heritable recombinant DNA, and regardless of the intended use, are considered to be new animal drugs by the FDA. Each time a gene is introduced into an animal, approval must be obtained even when the same gene is introduced into different animals. As for transgenic plants, it is recognized that the site of insertion of the animal's genome is difficult to control and may affect the level of expression of the transgene or the animal's native genes and therefore could affect the health of the animal. All animals containing a recombinant DNA construct are regulated, including subsequent generations of animals that contain the construct that were derived from the original manipulated parent by breeding the non-transgenic animal. A distinction may be made between transgenic animals that are to be used for food production and non-food transgenic animals. This lecture should be over in a couple of seconds now. The rest of it will continue in the next lecture. Transgenic animals that are used for non-food research products purposes or to produce human therapeutic proteins are contained within controlled environments for food production and may not be strictly regulated. Thank you. We'll continue in the next lecture.